Sleep functions as an active neurological state, contradicting the legacy view of unconscious dormancy. This pluripotent state possesses the capacity to manipulate and repair multiple distinct biological systems simultaneously. It is governed by a rigid, non-compressible biological architecture. To understand how this system operates, we will map this 8-hour architecture chronologically, from the initial transition to the final waking breath. We begin by examining the precise thermoregulatory triggers that dictate sleep onset latency. Sleep onset latency, or SOL, is the exact duration of time it takes a subject to transition from full wakefulness into the first stage of sleep. From there, we map the metabolic repair mechanisms engineered into deep non-REM sleep, the slow wave phase dominating the first half of the night. Finally, we uncover the specific hormonal releases and longevity-predicting mechanics of REM sleep, the active brain state characterizing the early morning hours. Mapping this progression reveals that sleep is a precise clinical intervention rather than an optional daily habit. Our timeline begins roughly 120 minutes before unconsciousness. This thermal map illustrates the core requirement for sleep, a steep decline in core body temperature. This chart displays the distal proximal skin temperature gradient, or DPG, vascular heat displacement to the extremities. As core temperature drops, DPG rises. We can manipulate this biological temperature gradient through a mechanical intervention known as passive body heating. Passive body heating involves applying external heat to the skin to trigger internal physiological changes. A 2019 meta-analysis by Hagayeg and colleagues demonstrated that water-based passive body heating at temperatures between 40 and 42.5 degrees Celsius significantly shortens the time to fall asleep. We observed the scale of this intervention in a 2021 cohort study by Tai and Associates, which tracked over 1,000 older adults engaging in the cultural practice of Japanese hot water bathing. The researchers mapped the timing of these baths, analyzing cohorts that scheduled their immersion strictly between 61 to 120 minutes and 121 to 180 minutes before their strict bedtimes. The physiological results show that this specific timing forced the DPG to increase by roughly half a degree Celsius in the critical 30-minute window prior to sleep. This thermal manipulation directly accelerated the neurological transition to sleep, reducing log-transformed actigraphic sleep onset latency by up to 0.32 log minutes. Actigraphic sleep onset latency is an objective measurement of sleep time derived from wearable motion sensors. Translating that logarithmic data into the practical patient experience, subjects saw a 14.8 to 16.5% reduction in their self-reported time to fall asleep. Precise. Applying aggressive external heat to the body triggers internal vasodilation, facilitating the core cooling required by the central nervous system to permit sleep. This hypnogram visualizes sleep onset, marking the commencement of rigid 90-minute physiological cycles. It plots distinct stages of sleep over time. The first half of the night is dominated by deep troughs, representing deep, non-rapid eye movement sleep. During this phase, hundreds of thousands of cortical cells within the brain synchronize, firing and going silent together in perfect biological unison. This synchronization produces a distinct electroencephalogram, or EEG, signature. An EEG records electrical activity in the brain, and in this phase, it exhibits high-amplitude, ultra-slow frequency brainwaves. While the brain slows, the body executes a systemic metabolic reset. Cardiovascularly, deep non-REM sleep acts as a natural blood pressure medication, dropping heart rate and easing vascular tension. Within the endocrine system, this slow wave state is critical for regulating insulin and stabilizing blood sugar. Depriving the body of this specific NREM phase generates severe clinical consequences for human metabolism. Sleep scientist Dr. Matthew Walker has quantified the cost of artificial NREM reduction. His data reveals a specific, measurable metric, dropping deep sleep by just 30%. Brainwave activity in specific cortical regions ramps up to 30% higher than when the subject is fully awake. To safely house this hyperactive cortical state, the brainstem initiates an evolutionary lockdown mechanism. A precise signal is sent down the spinal cord to the alpha motor neurons. Alpha motor neurons are large nerve cells responsible for directly initiating muscle contraction. This signal induces total voluntary skeletal muscle paralysis. Generating this REM architecture requires an extreme shift in chemical neuromodulators. REM is the only time in a 24-hour cycle where the neurotransmitters serotonin and noradrenaline are completely shut down. Simultaneously, levels of acetylcholine ramp up to drive this active brain state. Acetylcholine is a neurotransmitter heavily involved in memory and cortical arousal. Introducing external chemical disruption into this model highlights its fragility. Alcohol acts as a potent REM blocker. This blockage exacts a massive endocrine cost. Alcohol-laced sleep results in a greater than 50% drop in systemic growth hormone release. Suppressing this phase also prevents the peak release of testosterone, which natively occurs during REM cycles. The ultimate epidemiological consequence of depriving the body of REM is quantified in a machine learning analysis out of Harvard University. Researchers in the Beth Clemens group discovered that out of all sleep stages, REM is the strongest linear predictor of human longevity. They calculated a precise mortality metric. Every 5% reduction in REM sleep strictly correlates with a 13% increased risk of all-cause mortality. REM sleep operates as an irreplaceable biological mechanism tethered directly to human lifespan, hormonal stability, and mortality avoidance. This completed hypnogram charts the entire 8-hour sleep period as a single engineered biological system. This biphasic engine dictates that human function requires both the metabolic baseline established early in the night and the endocrine repair completed in the morning. This creates a structural trap for sleep deprivation. Artificially truncating total sleep time disproportionately destroys specific vital physiological phases. Delaying a bedtime and losing the beginning of the night destroys the NREM metabolic reset. Conversely, waking up early limits the REM cycle, undercutting hormonal balance and longevity. We are left with only one manipulable entry point, utilizing targeted thermoregulation like passive body heating to trigger the initial biological cascade. The data suggests that managing these 90-minute rhythms is not a matter of hacking rest, but of adhering to a biological requirement for metabolic and hormonal maintenance.